So a topic that I have also been wanting to discuss is the 5 modes of action of microbial drugs. The first mode of action would be the inhibition of cell wall synthesis. Now, the way this is actually done is most, if not all, penicillin and cephalosporin antibiotics have what is called a beta-lactam ring. Essentially, the beta-lactam ring will inhibit an enzyme called transpeptidase. Now, what transpeptidase does is it is the enzyme that is responsible for cross-linking the peptide chains between N-acetylmuramic acid. So peptidoglycan in gram-positive bacteria is composed of N-acetylmuramic acid and N-acetylglucosamine. And this is kind of the layer that makes up peptidoglycan, and then they are connected through peptide chains. So essentially all the beta-lactam ring does is it kind of blocks off the cross-linking between peptide chains. Because gram-positive bacteria rely heavily on their cell wall, they will succumb to osmotic lysis due to this. So they will kind of lyse or explode, implode on themselves due to these antibiotics. So they are very effective. They're obviously not that effective towards gram-negative bacteria just because gram-negative bacteria have a cell membrane on the outside as well. So it's really just meant for those gram-positive bacteria. Okay, the next mode of action would be the inhibition of protein synthesis. So these are drugs that directly affect the 70S ribosomes within bacteria. The reason these are effective is because bacteria and humans have different ribosomes. So bacteria has 70S, humans have 80S. So these are very effective because we won't experience any side effects from this. But there are 3 main kind of pathways or ways that these antibiotics work to inhibit protein synthesis. So the first way is that they can bind and inhibit peptide bonds. So I'm going to draw this out. Essentially here on your tRNA, you can see this is where the amino acid is going to kind of bind. And then it would get passed on, or I'm sorry, this would get passed on to here. For example, chloramphenicol is going to actually be one of those antibiotics that will kind of bind and inhibit other amino acids from binding, creating that long amino acid polypeptide chain. The other kind of way is, for example, tetracycline is, for me and my experience, tetracycline is the most popular protein inhibitory antibiotic. All this does is it prevents tRNA from actually binding or attaching to mRNA. So if you guys are familiar with how protein synthesis works, mRNA will go along the 70S ribosome, tRNA will attach to it, it will get pushed through the ribosome. So tetracycline will prevent these tRNA cells from actually binding to the mRNA in the first place. Streptomycin is another one. So this is something that's actually going to change the shape of the 30S ribosome, kind of distorting it. It's going to cause the mRNA codes to be read incorrectly. Again, no protein synthesis is going to be happening with that. So the third pathway would be the inhibition of nucleic acid synthesis. So this would prevent DNA, RNA replication and transcription. The way these work are they mainly target enzymes responsible for catalyzing and speeding up the reaction of DNA replication and or RNA replication. For example, this antibiotic here is responsible for freezing DNA gyrase. DNA gyrase is responsible for relieving supercoiling tension during DNA replication, but essentially this antibiotic will freeze it, so it won't relieve that tension. Ultimately, this causes replication forks to kind of stall, prevent the replication of DNA. Rifampin is the other popular antibiotic in inhibiting nucleic acid synthesis. So rifampin will overall just stop RNA polymerase from working. So this is going to prevent the transcription of DNA to RNA. So the fourth mode of action would be damage to the cell membrane. Now, the problem with this is that humans also share cell membranes. So the kind of loophole around this is by applying these antibiotics topically. If we were to ingest these antibiotics systemically, we would face a lot of side effects just because, like I said, if we're damaging the cell membranes of bacteria, us as humans, we also share cell membranes. It can cause a lot of harm to us if we do ingest them systemically. So topical use is preferred, and this isn't also the first line of defense against antibiotics, or I'm sorry, against bacteria. There would be more options ahead of this. Would be kind of towards the end of what you would be prescribed. But what these antibiotics will do, they will bind to the membrane's phospholipids, causing them to become permeable, the membrane will become leaky, so more ions, nutrients, metabolites, everything like that is going to kind of leak in and out. Polymyxins are good examples of this. Daptomycin is a good example of this. Fifth mode of action would be the inhibition of metabolites or metabolic processes. Usually act as competitive inhibitors for bacterial enzymes. For bacteria specifically, one of the most targeted pathways is the folic acid pathway. So humans get folic acid through nutrients, food like that. Bacteria have to synthesize their own folic acid. Without folic acid, bacteria won't be able to synthesize DNA. Proteins and stuff like that. These drugs often mimic normal bacterial metabolites. So what they'll do is they'll kind of trick the enzymes into binding with them instead of the real substrate. So overall, if they can't produce folic acid, they won't be able to synthesize DNA and amino acids and stuff like that. Yeah, so the 5 modes of action real quick: inhibition of cell wall synthesis, inhibition of protein synthesis, inhibition of nucleic acid synthesis, injury to plasma membrane, and inhibition of metabolite synthesis. I'll be doing a video on the modes of action for antiviral drugs, but I hope this was helpful.